everyone, this is Marika and welcome back to another session of Community Health Nursing. So in our previous few sessions, we have discussed the first unit in which we've seen what is community, what are the features of a community, what is community health, and we also discussed about community health nursing. We discussed about health and its dimension, the concept of health, and we have also discussed about health promotion and health maintenance. I hope you've understood the first unit clearly. And from this session onwards, we are going to start with the second unit, which is eugenics. Now, before we go and understand, before we have a discussion on what eugenics is and why as nurses are we studying about eugenics, we have to understand about the basic functioning of our body, the basic composition of a human body. So before we go on to study eugenics directly, let us understand the development in human body, how our cells forms, what are chromosomes, and then we will go and study about eugenics. We will have a discussion on what are chromosomes, how are they formed, and different cut, and genes and how are they formed, and why is it necessary for us to read or understand about genes and chromosomes right now? Because as we have a proper understanding about genes and chromosome, only then we will get to understand about the genetic disorders that we find. The concept of underlying genetic disorder was found or was understood only after having a study on chromosomes and genes. So we also as nurses have to understand about the structure, about the functioning and the number of chromosome in human body in order to have an understanding of genetic disorders. We are already aware that certain genetic disorders such as Down syndrome, Klinefelter syndrome, and Turner syndrome occurs due to the defect in the number of chromosomes, right? Genetic disorders may occur due to more number of chromosome or the location of the chromosome is changed or the genes and chromosomes plays a major role in affecting the health of an individual. It may lead to various chromosomal abnormalities and various genetic disorder. In order to make sure that we have a proper understanding about genetic disorder or chromosomal abnormalities in human being, we need to understand what are chromosomes, how are they formed, and what are genes as well. As I said, few of the disorders such as Down syndrome, Turner syndrome, Klinefelter syndrome and Turner syndrome occurs because of certain abnormalities in the chromosome. So earlier, there were no any kind of investigation for us to understand whether there were additional chromosomes or whether there were deletion of chromosomes. There were no investigation that would give us an idea about the abnormality in the chromosomes or in the genes. But now there are certain investigations that helps the healthcare professional to understand if there's any kind of abnormality, if there's addition, deletion, or any kind of shortage in chromosomes and in genes in the human body. So let us first understand what are chromosomes. In human being, as we know, there are 23 pairs of chromosome, 23 pair from the father and 23 pair In human being, as we know, there are 23 pairs of chromosome, which is 23 from each of the parent, 23 from the father and 23 chromosome from the mother. So there are 23 pairs of chromosome in any human body, 23 from the mother side and 23 chromosomes from the father side. Now, if we talk about chromosome, a chromosome can be defined as a K-shaped structure which is actually made up of DNA and proteins. There can be histone proteins and there can be non-histone proteins. So chromosomes are formed or it is made up of DNA and two different types of protein, that is histone and non-histone protein. The chromosomes are visible in the nucleus of the cell. So what is a chromosome? A chromosome is a K-shaped structure. As you can see here in the picture, a chromosome is a K-shaped structure and it is made up of various DNAs and two different types of protein, which includes the histone protein and the non-histone proteins. Now, where is the chromosome located? As I said, chromosome is visible in the nucleus of the cell. So the chromosome is located inside the nucleus of the cell. In a human body, there are two different types of cell. As we know, we have somatic cells and we have germ cell. In somatic cell, there is a cell division called as mitosis. So in somatic cells, we have mitosis that takes place. During the time of mitosis, which is a form of cell division, the human body, the cells in the human body divides into daughter cells, or they form daughter cells with equal number of chromosomes in both the daughter cells. Mitosis takes place in somatic cells, and from somatic cells, the daughter cells that we get will have the same number of chromosome as the mother cell. But as I said, the different type of cell, that is the gamete cells or the germ cells, undergo meiosis. Meiosis is also a form of cell division, and during the time of meiosis, the daughter cells will only have half number of chromosomes. So there are two different types of cells, somatic cells and germ cells. Somatic cell will undergo mitosis and it will form, or it will create daughter cells with the same number of chromosomes like the mother chromosome. Whereas in gamete cell or the germ cell, it undergoes meiosis, which is a form of cell division, and after meiosis, the daughter cells will not have the exact number of chromosome that the mother cell had, but it will have half of the number. Somatic cells are the cells in our body that are other than the gamete cells or the germ cell, which includes the ovum or the sperm cell. All the cells in our body that are not germ cell can be referred to as somatic cells. Now, when we talk about somatic cells, as I said, they undergo mitosis, right? When they undergo mitosis, they form daughter cells, and daughter cells will have same number of chromosome like the mother cell. So we can say that somatic cells are diploid. What is the meaning of term diploid? The term diploid means they contain two sets of chromosome, one inherited from each parent. So a somatic cell undergoes mitosis, it forms the daughter cell. In the daughter cell, we have equal number or same number of chromosome like the main mother cell. Now, the chromosomes that are present in the daughter cells are two sets of chromosome, one inherited from each parent. Chromosomes are different, they vary in their shape and also in their size. Depending upon the size of the chromosome, the chromosomes are divided into various groups such as A, B, C, D, E, F, G and sex chromosome. So chromosome can vary in shape and size, and according to the difference in the shape and size, chromosomes are divided as A, B, C, D, E, F, G and sex chromosomes. Now, when we talk about the sex chromosomes, we know that in female there are two sex chromosomes and in male there are two sex chromosomes. But one important thing to remember here is female have two sex chromosomes which are same, which are the exact same, whereas male have two sex chromosomes which are different from one another. As we know, in female we have X and X chromosome, whereas in male we have X and Y chromosome. The X and X chromosome in the female is called as homogenetic sex, whereas the X and Y chromosome in the male is called as heterogenetic sex. We already know what is homogeneous and what is heterogeneous. The same kind of group, the same type is called as homogeneous, whereas if there is two different kind or if there are different kinds, then it is called as heterogeneous. In female we have X and X chromosome, therefore in female we have X and X which is similar to one another, which is exactly the same. So we call it as homogenetic sex, whereas in male we have X and Y, therefore we call it as heterogenetic sex. I hope you've understood about chromosomes so far. A chromosome is a case-shaped structure that is present in the nucleus of the cell. Chromosome is made up of various DNA and two different types of protein, that is non-histone proteins and histone proteins. Now, during a time of cell division, we can consider that we have two different forms of cell, that is somatic cell and germ cell. In somatic cell, the cell division that occurs is mitosis, and after mitosis, the daughter cells will have the same number of chromosome. Whereas in meiosis, the daughter cells will not have the same number of chromosome but the half number of chromosomes. Chromosomes vary in shape and size, and according to the difference in the shape and size of the chromosomes are divided as A, B, C, D, E, F, G and sex chromosome. When we talk about sex chromosome, we know that in woman we have two sex chromosome and even in male we have two sex chromosome. But in woman it is homogenetic sex, which means both the sex chromosome are X and X chromosome, which means the male sex chromosome has the X and Y chromosome. Now let us see what is a chromatin. A chromatin is the combination of DNA and protein. So chromosome is formed by chromatin. So before chromosome is formed, we have chromatin. What is a chromatin? It is a combination of DNA and protein. So chromatin are the basic structure of chromosome. Chromatin are formed by combining DNA and protein, basically the histone proteins, and the condensed chromatin forms the chromosome. For the chromosomes to be formed, we need chromatin. What is chromatin? It is the combination of the DNA and the proteins. It can be histone proteins basically and even some non-histone proteins. And when it combines, it forms a condensed chromosome. When it combines, it forms chromosome. So we can say that chromosomes are condensed chromatins. As I said, the chromosomes are condensed chromatin. When we take out the DNA and when we examine the DNA, it is more than 2 meter long. The length of the DNA is around 2 meter. Therefore, it has to be condensed in order to form or in order to fit inside the nucleus, right? So there is condensed chromatin. So as I said, a chromosome is a K structure, and you can see here in the picture, there is something in between that is holding the chromosome, right? That is holding the chromatids. The structure that holds the two chromatids are called as centromere. So you can see here there is a chromosome, and chromosome is formed by chromatin. You can see a structure that holds two different chromatids. That structure that holds two chromatid is called as centromere. Here in the screen, I have displayed a chromosome. The arms of the chromosomes are called as chromatids. So there are sister chromatids and there is a centromere. What is the centromere? The centromere holds the chromatids and forms the chromosome. So a centromere is a structure that holds the chromatids. Depending on the location of centromere, there are three different types of chromosome. The first type of chromosome is called as metacentric chromosome. Now from the term itself, we can understand a metacentric chromosome is the chromosome which has the presence of centromere exactly in the center, dividing the chromatids into two equal arms. Here on the screen, I have displayed a picture of metacentric chromosome. As I said, a metacentric chromosome is a chromosome that has the presence of centromere in the center or in between, and it divides the arms of the chromosomes into two equal halves. The upper half of the chromosome is called as P arm and the lower half of the chromosome is called as Q arm. In metacentric chromosome, the centromere is present exactly in the center, dividing the chromatids or dividing the arms of the chromosomes into two equal halves. The second type of chromosome, according to the location of centromere, is submetacentric chromosome. Again, from the term itself, we can understand that in submetacentric chromosomes, the centromere is not present exactly in between, but it is slightly away from the center. Again, here in the picture, you can see that the centromere is not exactly in between, but it is slightly off or away from the center. Here, obviously, because the centromere is present slightly upward, the P arm is shorter than the Q arm. So it does not divide the arms of the chromosomes. Here we get short P arm and a longer Q arm. The last type of chromosome, according to the location of centromere, is echocentric. What is the meaning of echocentric? Here, the centromere is located towards the end of the chromosome. It is located towards the end of the chromosome. So in metacentric, we had the centromere exactly in between. In submetacentric, the centromere was present slightly off from the center. And in echocentric, we can see that the centromere is present towards the end of the chromosome. Again, here on the screen, you can see the presence of centromere is towards the end of the chromosome. Therefore, it is called as echocentric chromosome. Now here on the screen, you can see three different types of chromosome. The first type of chromosome is a metacentric chromosome wherein the centromere is present exactly in between, dividing the upper and lower arm, that is the P and Q arm of the chromosomes, into two equal halves. The second one that we have on the screen is submetacentric, wherein the centromere is not present exactly in between but slightly upward, dividing the chromosome into shorter upper half and a longer lower half. And the last one we have is the centromere is present towards the end of the chromosome, I hope you've understood what is a chromosome, what is chromatin, and what is centromere, and how according to the location of centromere we get three different types of chromosome. Now that we've understood what are chromatins, let us see what is the function of chromatin. The very first function of chromatin is to pack DNA into smaller or compact form. So as I said, DNA, the length of the DNA is around 2 meters, which is very long. Therefore, the DNA has to be formed. The DNA has to be in condensed form in order to fit inside the nucleus of the cell. The chromatin helps to condense the DNA and therefore make it very small to fit inside the nucleus of the cell. That is the very first function of chromatin. Chromatin also strengthens the cell for any kind of cell division, that is also for mitosis and meiosis. Chromatin helps to strengthen the DNA for cell division, either for mitosis or for meiosis. And the last function of chromosome is to serve as a mechanism of gene regulation. So chromatin is also involved in gene regulation, which is the combination of DNA and proteins, majorly stone proteins, and chromosomes are made up of chromatin. Chromatin helps the DNA and the protein to be condensed so that it can fit inside the nucleus of the cell. Let us now see what are genes. Genes refer to inheritable traits. The basic unit of heredity passed from the parent to the child or to the children is called as gene. Now, genes are also made up of DNA and they are arranged one after the other in a sequence within the chromosome of the cell. Okay, this is very important to understand. Chromatins forms the chromosome, and inside the chromosome, genes are located. What are genes? Genes are the inherited traits. Okay, so genes are located inside the chromosomes, and chromosomes are formed, or chromosomes are made up of combination of DNA and proteins, which are the chromatin, which can be termed as chromatin. Now, genes are located in a particular place inside the chromosome and they are arranged in a proper sequence. A gene will contain, or genes contain, the information about specific protein that leads to expression of particular physical characteristic in an individual. So as I said, gene is the inheritable trait and it is located inside the chromosome in a specific location and they are arranged in a sequence, right? So gene will have the information about a certain protein. Okay, now this protein will express a characteristic in an individual. For example, the color of the hair, the color of the eye of an individual, the color of the hair or the color of the eye of an individual. So this protein will contain information about the physical characteristics of the individual. Each person has two copies of gene, one inherited from each of the parents. So every person will have two copies of gene and one of the gene will be inherited from one of the parent. Most of the genes in all human beings are the same. In almost all of the people, it is the same. But there's only 1% of genes that are different from one individual to another. That 1%, that small difference of the gene in each of the individual contributes to the unique characteristics and unique feature of an individual. These are called as alleles. Okay, so individual will have two copies of gene and each of these genes come from each of the parent and these are called as alleles. The same kind of alleles can come in the individual from the parent, or two different kind of alleles may be inherited from each of the parent. If the alleles that is inherited is different from one another, only one of the allele will be expressed. Alright, so from each of the parent we will receive the alleles. If this alleles that we receive from our parent is different from one another, then only one of the allele will be expressed. The one that will be expressed is called as dominant gene, and the one that will not be expressed or that will be masked is called as recessive gene. So it is very important for us to understand, therefore I'm repeating it again, from our parents we receive two copies of gene, or we can say two versions of genes from each of the parent. Now these versions of genes are called as alleles. These alleles can be similar to one another or they can be different from one another, because we are inheriting it from two different people, right? Mother and the father. Therefore, we can get two different type of allele or we can get the similar allele. If we get two different allele, only one of the characteristics will be expressed. The characteristic that will be expressed is called as dominant gene, and the one whose effect will be masked or whose effect will not be seen is called as recessive gene. We have two different type of gene. One is dominant gene and other one is the recessive gene. The dominant gene is the gene that has the expression cut. The dominant gene is the gene that will be expressed, whereas the recessive gene is the gene that will not be expressed. It will be present, but it will not be expressed. So that is it for today's class. To summarize, today we have discussed the basic formation of human being, which includes the chromosomes, chromatids, and genes. We've seen what are chromosomes, how are they formed, what are they made of, what are chromatids, what are their function, what is a centromere, where are they located, and how it gives rise to different kind of chromosome. And we also discussed about genes and what is the function of gene in human body. In our next session, we will discuss about chromosomal abnormalities and genetic disorders. I hope the class was helpful and I'll see you all in the next session. Thank you and have a great day ahead.